Starting, all attendees are in listen-only mode.
Hello everyone and welcome. Thank you for attending today's webinar, Optimizing Calcium and Phosphorus Contribution in Skeletal Integrity Through Phytase Use in Broiler Diets. Today's webinar is sponsored by DuPont and presented by Watt Global Media. My name is Terrence O'Keefe. I'm the editor of Egg Industry Magazine, and I'll be your moderator today. I'd like to let everyone know that we will be having a question and answer segment at the end of our presentation today. Please feel free to submit your questions during the presentation. You have a function for entering them in with your keyboard. Your submissions will remain anonymous to the rest of the audience and will only be visible to the moderator and speakers. We will answer as many questions as we can with the time allowed. I'd like to note that today's webinar is being recorded and will be available to view as an archive. If you would like to watch the archive of this webinar or share the archive with some of your coworkers, they can go to www.wattagnet.com/webinars.aspx. Also, at the end of today's webinar, there'll be a brief survey. We would greatly appreciate if you took a few minutes out to fill out the short 8-question survey. We do have a number of webinars coming up on our schedule. Matter of fact, this week we have 2. On Thursday, April 10th, we have a webcast, Consumer Trust in Poultry, Bridging the Gap Between Where We Are and Need to Be, sponsored by Zoetis. On Friday the 11th, we have Grain and Meat Outlook webinar number 1 for 2014. This will talk about the prospects for the crops this year and for meat and poultry prices in 2014. And finally, on May 7th, we have another webinar, You Never Have Time to Recover from a Good Start, The Importance of Early Flock Performance and Recent Innovations in Hatchery Probiotics, which is sponsored by the Pacific Vet Group. To register for any of these upcoming webinars, please visit us at wattagnet.com. Today's webinar is being brought to us by DuPont. DuPont has been bringing world-class science and engineering to global marketplace in the form of innovative products, materials, and services since 1802. The company believes that by collaborating with customers, governments, NGOs, and thought leaders, we can find solutions to such global challenges as providing enough healthy food for people everywhere, decreasing dependence on fossil fuels, and protecting life and the environment. For additional information about DuPont and its commitment to inclusive innovation, please visit www.dupont.com. Our speakers today are Dr. Douglas Corver and Dr. Peter Plumstead. Our first speaker, Dr. Corver, holds BS in Agriculture from the University of Saskatchewan, Master of Science from the University of Delaware, and a PhD from the University of California at Davis. In addition to undertaking practical research on feed quality and supplement evaluation in poultry diets, he is also focused on nutrition, immune function, interactions, and has a specialty in poultry bone biology. He has had over 45 peer-reviewed articles published in journals and has achieved over 570 peer-reviewed citations. Dr. Corver currently teaches introductory animal nutrition and poultry nutrition courses at the undergraduate level and is a co-instructor for graduate level courses in advanced animal metabolism and advanced feed science and technology. Dr. Corver?
Thank you for the introduction, Terrence. There has been continued growth in the poultry industry. Worldwide demand is increasing, and we expect that demand to continue to increase. Projections suggest that there's a 35% increase, and by 2020, we expect to see poultry, particularly broiler meat, as the #1 worldwide protein source. With that increasing demand comes an increased need for production, and a lot of that production will be met by increasing bird numbers, but there's continual demand for increasing growth rate and increasing efficiency. That rapid growth rate, however, can put pressure on metabolic systems, including the skeleton. And during the 1980s and 1990s, I think the geneticists were probably paying more attention to growth rate and efficiency and not as much attention to metabolic disease, not as much attention to skeletal health as they should have. In recent years, the primary breeders have done an excellent job of selecting for both growth rate and metabolic health, including skeletal health, but I think that rapid growth rate really has put the chicken on the edge metabolically, and so although we don't tend to see a lot of problems right now, I think the potential is there. As we continue to select for increasing growth rate, I think the broilers may actually be getting closer and closer to a physiological limit of what they're capable of. So to put it all in context, if we're going to understand how to prevent these skeletal issues, how we can optimize calcium and phosphorus metabolism, and how we can best use phytase to our advantage, we need to understand the process of bone formation. Bone development really begins in the egg during embryonic development, and it initially starts with a cartilage matrix. You can see this initial model made exclusively out of cartilage, and at about 10 days of incubation we start to see mineralization of the bone, and in this figure you can see the red stained area is mineralized bone, the blue area is organic matrix. And we can see that as the bone develops in the embryo, we see increases in size, but also increases in mineralization. Post-hatch, the same basic process takes place as well. We have the development of the cartilage frame that initially takes place in the embryo. We have the formation then of an organic matrix that is mineralized starting at the mid-region of the diaphysis. That is a process that continues on from the embryonic development. The mineral that's deposited is primarily hydroxyapatite, and the chemical formula for that has calcium and phosphorus in about a 2-to-1 ratio. And this is by far the main use of dietary calcium for both growing birds and even birds in maturity. And that 2-to-1 ratio of calcium to phosphorus in hydroxyapatite really is the basis for what we typically consider to be the calcium to phosphorus optimum in the diet, of that 2-to-1 ratio. Obviously, there's a substantial growth in body size as the chick hatches and progresses through to market weight. Both the tibia and the femur have approximately a 4-fold increase in length over the 6 weeks, approximately 6 weeks of the broiler's life. And the basic process is as I described on the previous slide. So we have initially the formation of an organic matrix, the calcification of that matrix beginning at the mid-region. Now the growth in length takes place at either end of the bone, and these long bones have a growth plate, and the growth plate lays down new cartilage, and the cartilage is deposited in this growth plate region, and there is a progression of cells and cell development as the bone elongates. Once we look closer to the mid-region of the bone, the diaphysis, when we look at this metaphysis, we have a region where those original cartilage cells and cartilage structures start to degenerate and degrade. This is part of the normal process of calcification. So in the degenerating zone, the calcifying zone, we see a change in the type of cell that are present and the degeneration of the cells, and that then becomes mineralized. As the growth proceeds, we then have an uptake of bone from the resorption zone that can then be used to elongate the bone. Now, when we look at bone growth, we also have to look not just increases in size, we also have to look at increases in mineralization. And we can see from this work done by one of my graduate students, Mike Leslie, we can see that over time there is an increase in cortical bone density. Now, this is measured using quantitative computed tomography, and we can see approximately about a 50% increase from hatch to 6 weeks of age in bone density. And this is a natural progression as the bones become more mineralized, as the deposition of calcium and phosphorus in hydroxyapatite becomes more pronounced. And this was confirmed in some recent work that we did in my group. In a different flock of birds, obviously, at about 6 weeks, we saw roughly the same cortical bone density. What's interesting is when we look at broiler breeders, which are much more slow growing, we have a substantially higher cortical bone density in breeders than we do in broilers. And there's a couple of reasons for this. First of all, broiler breeders are slower growing, and they have more time to mineralize because they have a longer life cycle. Now, so far we've talked about bone growth in terms of increases in bone length, but we also obviously recognize that the bone is increasing in size, in width. And we can measure this by looking at the cross-sectional area. So if you can visualize, we're taking essentially a slice from the mid-region of the bone, and we're looking at the cross-sectional area. And as the broiler grows, what we see is not only an increase in mineralization in terms of cortical bone density, but we see a substantial increase in bone cross-sectional area. So bones are growing rapidly in length, but they're also growing more rapidly in cross-sectional area. And it amounts to about a 30-fold increase between hatch and 6 weeks of age. When we look at broiler breeders, we see that the overall cross-sectional area is smaller than in broilers. And this is interesting because typically broiler breeders are going to be heavier than broilers, but that slower growth rate changes how the bone is deposited. Now, if we look at early in life, if we look at bone formation, we have a situation where the bone cross-section is essentially a circle. We have the outer region, or the periosteum region. We have the cortex of the bone, and then we have the marrow cavity. Normal bone formation normally proceeds along canals that are deposited in the bone, and these canals gradually become mineralized. And with slow growth rate, we have the deposition of calcium into these canals, these Haversian canals, and these form osteons. And these are the basic structure of the bone. With rapid growth rate in the broilers, however, we have to think of bone as a structural material. And when we look at bone as a structural material, what we see is that the bone is stronger when there's a dramatic increase in the outer diameter of the bone, even at the expense of not filling in all those canals, all those osteons. And so in broilers, what we get is typically a porous bone surface. Now, we have to remember that we market broilers at 6 weeks of age approximately, and in some markets even earlier than that, and the bone is an immature structure. We're marketing these birds at a very young age. If we consider that sexual maturity occurs at about 25 weeks of age, we are marketing these birds at a very, very young age. And so this porosity is a function of the very rapid growth rate of modern broilers. And as long as we are going to market birds at such young ages relative to sexual maturity, we're probably going to have to deal with this bone porosity issue. If we were to allow broilers to achieve a more mature body weight, or if we look at broiler breeders, what we see is that over time, these canals become filled in and we have a much more solid structure. And so this explains the higher cortical bone density that we saw in the previous slide. So again, it's important to underline that the bone development of broilers is really a function of the age at which we market birds. So we are marketing immature birds. It's unreasonable to expect them to have a mature bone structure. Now in the past, the rapid growth rate greatly increased the risk of skeletal deformities. In the 80s and 90s, again, skeletal problems were a big issue. We saw the emergence of lighting programs and feeding programs that were essentially designed to slow down early growth rate in order to reduce bone deformities such as tibial dyschondroplasia, angular deformities, and a number of other bone problems. I think the primary breeders have done an excellent job recently of selecting not only for rapid growth rate, for breast meat yield, and for efficiency, but they've also paid a lot of attention to skeletal health and metabolic health, and I think that has certainly had a positive effect on the skeletal health of modern broiler chickens. However, as we continue to select birds for more rapid growth, we have to remember that we are still dealing with an immature bird, and as the birds grow faster, that immaturity or that relative immaturity is likely to increase at market weight. So we have a situation where we have a high degree of bone porosity. The chicks are not able to fill in all those canals, all those osteons, and still maintain a strong bone structure. The strategy is to increase bone strength by increasing diameter even while bone porosity remains high. Now we can look at bone porosity, and there are some consequences at processing. There may not be consequences in terms of the function of the bone, in terms of the ability of the birds to walk around. But if we look at the bone porosity, what we see, this is a femur, a cross-section of a femur of a 6-week-old broiler, and we can see that the pressure, the loading pressures on the bone lead to a non-concentric circle type of appearance. So we have a region on one side of the bone that's quite thick, but you can see the pores. On the opposite side of the bone, we have a region that's very, very thin, but very highly mineralized. When we take a closer look at this region, we can see that these pores exist, and some of the pores actually extend from the marrow cavity up at the top right of this picture all the way down to the periosteum. And so what we have is a situation when the birds are processed, and particularly when the meat is frozen with the bone in and then cooked, the expansion of the marrow during the freezing process causes the leakage of pigment out of the marrow cavity into the meat adjacent to the bone, and we can have this problem with black bone. We've done some work with this, and this is not a defect from the standpoint that it is unwholesome or is a risk to consumers. However, many consumers do not appreciate the appearance of this dark staining of the meat next to the bone. Now, because we have modern birds with rapid growth, and I think that rapid growth at the present time is not necessarily causing major problems for the industry, but I think we're approaching physiological limits to bone formation. We seem to be putting the birds in a situation where the dietary calcium and phosphorus content may be becoming more and more of an issue. Again, as I think we're approaching biological limits. In the past, we've measured bone metabolism, or we've done studies with bone metabolism, typically using destructive measures of measuring bone quality. So, for example, we sample a proportion of the birds from a research flock. We dissect out bones, and we look at things like bone ash, measuring the mineral content in general. We can measure the content of specific minerals such as calcium and phosphorus. We can measure bone breaking strength. Now, these have been effective measures. We've learned a lot about calcium and phosphorus metabolism and bone formation in broilers because of these techniques. However, they are destructive. And one of the exciting things I think that's happening in this area of research is the development of non-destructive techniques, typically using imaging technology. There are a number of different imaging technologies that have been used. I think one of the most relevant or most important developments in the genetic selection programs of the primary breeders for increased skeletal health has been the use of the lixoscope, allowing the breeders to look at bone development in live birds. and that has had a great impact on the skeletal health of birds. There are other technologies such as DEXA, or dual emission X-ray absorptiometry, also ultrasound. And I think these new technologies are important because if we're pushing these birds closer to physiological limits in terms of bone formation, I think we need to really have an excellent handle on how those bones are forming. In my research group, we use two techniques. The first one is quantitative computed tomography. And the computed tomography is essentially a three-dimensional X-ray. And so what this does is allows us to, in live birds, look at essentially cross-sections of bones. We can do this in live birds. We don't need to sacrifice the birds. And this is a very powerful tool because we can look at development of the bone over time in an individual bird. And this shows us regions of higher bone mineralization and regions of lower bone mineralization. We can look at the development of bone mineral over time. A second and related technique is microcomputed tomography, and essentially this is a similar technology. The resolution is just much, much higher, and in recent years we've had the advent of microcomputed tomography units that allow us to scan live birds. In the past, because of higher X-ray doses and small chambers, we've had to focus on particular bones. We now have the technology that we can use this in live birds. So most bone research has focused on calcium and phosphorus because they are by far the most prevalent minerals in bone, and therefore they're the most prevalent minerals in diets. And this is true not only of rapidly growing birds, but also birds at maintenance. The second reason is that in general, the calcium and phosphorus requirements, or the calcium and phosphorus supply that we provide in the broiler diets, have a much narrow range of appropriate levels because of that calcium and phosphorus interrelationship. We see that when there's an excess of calcium or phosphorus, we will cause a pressure on the other mineral because an excess of one of calcium or phosphorus is excreted as a calcium-phosphorus complex. So if we have excess phosphorus, the body tries to get rid of it by excreting it with calcium. If we have excess calcium, the body excretes it in the form of a calcium-phosphorus complex. So that balance, and not just the level of calcium and phosphorus, are extremely important in proper bone mineralization. There's increasing economic and environmental pressure to reduce the level of phosphorus in the diet. Phosphorus supplementation is becoming more and more expensive. There are concerns with phosphorus pollution, and so we've seen a drive to lower the calcium and phosphorus levels in the diet. And because of this, they have the opportunity to use phytase. Now, with the use of phytase, and my colleague Dr. Plumstead will be talking much more about this, and so I won't get into it too much, but we need to understand that phytase is an enzyme that works on substrate, and we have to have reasonable expectations of what phytase can do for us. And as long as we're using phytase properly, this can be an excellent tool to reduce the cost of our diets, to reduce phosphorus pollution, and maximize bone formation. So the conclusions, I think we need to remember that broiler chickens grow very, very rapidly, and when they reach market weight, they are very immature birds. As long as we continue to market birds at the size and age that we do, we will be marketing birds with immature skeletons. And I don't know that there's anything nutritionally that we can do to have those birds achieve a mature skeleton by 6 weeks or 5 weeks. I think what we need to do is we need to optimize the development within the constraints of biology. I think the primary breeders have done an excellent job of selecting for bone health and metabolic health as well as growth rate. I think we see a lot fewer skeletal problems than we did in the 80s and 90s. However, there doesn't appear to be an end, at least within the next 5 to 10 years, in the increase in growth rate. And I think as we continue to increase growth rate, we're going to see more and more pressure put on the ability of the broilers to mineralize the skeleton adequately before slaughter. and recognizing that adequately doesn't necessarily mean achieving a mature skeletal frame. The advent of new imaging technologies to look at bone metabolism in individual live broilers, I think, has the potential to really advance our understanding not only of bone biology, but how nutrition can optimize bone development. And as long as there's economic and environmental pressure to reduce phosphorus in the diets, we need to be very, very careful about the calcium to available phosphorus ratio. And I think a big part of that is the proper application of phytase. And my colleague will be talking more about this, but we need to consider the proper phytase matrices. We need to consider the phytase activity. And we need to remember that in order for phytase to work properly, there has to be the substrate available, or the phytate available. And with that, I thank you for your time and your attention.
Thank you for your presentation, Dr. Korver. Our second speaker, Dr. Plumstead, holds Master's and PhD in Nutrition and Poultry Science from NC State University, where his primary research focused on amino acid and phosphorus nutrition of broilers and broiler breeders. Prior to joining academia, Dr. Plumstead was engaged at Danisco Animal Nutrition, a part of DuPont, where he was responsible for application research and development of enzyme products for pigs and poultry. In addition to his work with enzymes, Dr. Plumstead has several years of commercial experience in the feed industry, having worked as a senior nutritionist within the Meadows Feed Group in South Africa. Dr. Plumstead? Dr. Plumstead?
Unmuted.
Great. Thanks very much, Terence. I appreciate the introduction. Let me just, okay, good. I think we're ready to go. What I wanted to talk about was follow on from what Dr. Korver was saying about the importance of calcium and phosphorus and its absolute role in bone mineralization and broiler performance, and specifically take a look at how phytase application can help us understand factors that impact phytase efficacy and use. Sorry, let me just make sure I get, okay. So if we first start off by taking a very simplified look at phytase, phytate and phytase, I think all nutritionists will be well aware that all feed ingredients of plant origin contain phytate phosphorus that is generally not very well digested by the animal. And so in a simplified form, we add phytases to increase availability of phytate phosphorus, and that in turn reduces the amount of inorganic P that is needed in the diet and saves money. However, one cannot really look at phytase in such a simplistic form simply because there's a lot of factors that actually affect dietary phosphorus digestibility as well as the absolute phosphorus and calcium contribution from phytase. And these can really be divided into three separate sort of categories. The first on your left-hand side is obviously the source of phytase selected and the dose of phytase that is selected for the operation, along with the methodology that is used to actually derive the phosphorus values or the phosphorus matrix of those phytases. A second category of factors would be things like dietary calcium level and the solubility and particle size of that calcium, and obviously associated also with vitamin D that affects the calcium absorption. As Dr. Korver mentioned, the substrate level phytate source is absolutely important as well, as is bird age, digestive passage rate, and health and disease status. So if we start off by first of all looking at phytase sources and methodology to derive matrices, what really stands out is that every single phytase supplier will be providing a matrix value for their product in terms of either digestible or available phosphorus, as well as a calcium matrix value. And if you put all these side by side in terms of 500 FTU equivalents, what becomes apparent is that the ratio of digestible phosphorus to available phosphorus that is recommended by suppliers seems to differ substantially, depending on what phytase source it is. And that's almost not possible. And what's critical, therefore, for nutritionists to ask when they select a phytase is how were the phosphorus matrix values determined? How does the phosphorus system that is recommended by the phytase compare to your own ingredient phosphorus matrix, and what about calcium matrix values for phytase? And then one also needs to understand the multiple factors that affect the variation in response to phytase, and hopefully we'll be dealing with a few of those today. So the first point is that digestible P is not the same as available phosphorus. So when looking at a phytase matrix, it's absolutely critical to distinguish between the two. And you can really see the differences in terms of inorganic phosphorus sources if you look at either the availability coefficient. So MCP is typically set as a benchmark of having availability of 100%, monodicalcium phosphate around about 98%, and dicalcium phosphate 95%. The digestible or retained P coefficients that were published by the CVB for those respective sources are 81, 79, and 78%. And that gives you a rough ratio of digestible P to available P of approximately 0.81 or 81%. So going back to calculating that ratio for different phytase sources, you can see that some phytase sources have got it right and others seem to be very conservative. Then moving on, what's also important to understand is that most phytase suppliers actually utilize a methodology called available P relative to an inorganic phosphorus standard to determine their matrix values. And quite simply how this works is to measure the tibia ash response in young broilers in response to added phosphorus from monocalcium phosphate typically, determine the response curve, which is a blue line here, and then in the same diet as the negative control, there's typically one or more levels of phytase added that is interpolated then with the response of the MCP curve or the inorganic phosphorus curve. So what that means is that the available phosphorus matrix is actually pretty much a total P contribution relative to monocalcium phosphate, and obviously will vary from study to study and be affected by the conditions of that study, such as the dietary calcium level. Then other phytase companies utilize what's called a digestible P system to determine their phytase matrices, and that works by measuring the response in ileal phosphorus digestibility to analyzed phytase in the diet. And it's a real simple way of determining a matrix, namely that you first determine your ileal P digestibility either in a negative control diet or the diets with added phytase. You then multiply that ileal P digestibility by the analyzed phosphorus value in the negative control to get an absolute amount of P that is absorbed in gram per kilogram of feed. And the phytase matrix in digestible P is simply the difference in the gram per kilogram P that is absorbed from phytase compared to the negative control diet. And those numbers are then typically to an available P by using a fixed factor. In this case, 0.91 or 1.09 is the coefficient that was used. So digestible P or available P, it doesn't really, I don't think it matters which one one uses, but what's important is that there are substantial differences in how these are determined. And the other important thing to understand also in some recent work that I've been involved in from the University of Maryland is that when available P from phytase, which is represented basically in the same way, namely the MCP response curve, and then two phytase doses, either 500 or 1,000 units, when that is compared to digestible P from determined on an ileal basis, as I just described, one can clearly see that the available P number that was determined in this study of 0.14% from 500 units was substantially higher, namely about 27% higher than the increment in digestible P. So the key point is that available P or NPP equivalents from tibia ash will always be higher than the equivalents determined in digestible P, and so the two should definitely not be mixed up, and they're certainly not the same. And so basically the take-home message from that section is that nutritionists really need to understand what research is underlying phytase matrix value and how this relates to the ingredient phosphorus matrix, whether or not one is based in the US and using the NRC available P matrix, or for example the French AP matrix, the Brazilian tables or the Dutch retained matrix. So the take-home message really is that nutritionists need to be very critical and ask the right questions to phytase suppliers in terms of how were matrix values determined in the first place before these are simply accepted as fact. Then moving on, the next thing I want to focus about is what about the effects of calcium, and specifically calcium on phytate hydrolysis and phytase efficacy. There's a lot of work that was done early on in the late 90s and early 2002 that showed that dietary calcium has a fundamental impact on phytate hydrolysis. This work published by Taman and Angel clearly shows this, namely that if you have a high calcium diet of 0.69% calcium and you reduce that to 0.17% calcium, you get a huge increase in phytate utilization at ileal level, and this is in the absence of phytase. And the reason for that big effect basically of calcium on phytate hydrolysis can be seen here, where at a high pH, in other words in the small intestine of the animal, as soon as the digester enters the duodenum, calcium starts precipitating with phytate, and it's not just calcium but also divalent minerals obviously, but because of the high concentration of calcium in the diet, this has a profound effect on phytate utilization in the lower intestine as a consequence of phytate precipitation in that area. And this can really be visualized very nicely again by this data from Dr. Angel, who clearly showed that effect, namely as soon as your pH increases above 4 and you have a solution of calcium and phytate in the actual flasks that we're showing here, you can see precipitation of that phytate. And that precipitation of calcium phytate complexes increases almost linearly as the pH increases further. So again, the key thing is that if your phytate has not been hydrolyzed by the small intestine, there's a high likelihood that calcium is going to start binding it, it's going to precipitate out of solution, and as a consequence your phytase will not be able to access it as well. And so with that in mind, there's obviously large differences between phytase enzymes in terms of their actual pH optima and what area of the digestive tract they're going to work on. This is some of the data that was shared with me by DuPont Laboratories back in 2012, where we can clearly see that something like a new generation Bullyoxella phytase has a much higher relative activity in the acidic portion of the digestive tract as opposed to either an E. coli phytase, which is shown by the green line, the Citrobacter phytase by the red line, and then also we're comparing that basically with a fungal phytase or an older generation Penniphore lysine phytase. So the take-home message is based on these differences in the relative activity of phytases in different segments of the digestive tract, there are going to be substantial differences in terms of where they work and also how they are influenced by calcium. And that was also shown previously back in 2004, again by some work by Taman et al. And what they did is they looked at in vitro phytate hydrolysis from phytase, and I'm just going to use a 60-minute example. So what you can see is there's two different phytase sources. The one was a bacterial phytase source, the second one was a fungal phytase source, and we're looking at the influence of calcium level on phytate hydrolysis either at a low pH 2.5 or a higher pH of 6.5. The key message is obviously that some phytases, namely the bacterial phytases, are able to work at low pH. The fungal phytases are not able to work at that pH at all. And also, when you look lower down at a higher pH, at 6.5 namely, there's also differences between phytases in terms of how these are affected by calcium. So again, take-home message is not all phytases are going to work in the same area of the digestive tract, and secondly, not all phytases are going to be influenced by calcium at that pH in the same way. There's also this other in vivo work that was done by the same group back in 2004 that showed exactly the same thing, namely in diets without phytase there was a severe negative effect of added calcium on phytate hydrolysis and P digestibility. But we're also seeing differences between phytase sources in terms of how well they were able to increase ileal P digestibility in vivo. So again, effects of calcium and phytases are not going to be the same. And then in some of my own work that I did back in 2010 at North Carolina State University, we showed similar things, namely that too high levels of calcium can be severely detrimental when it comes to ileal phosphorus digestibility. And this study was done with an E. coli phytase at what I would call a low dose for today. So 500 units of E. coli phytase was severely inhibited in terms of ileal P digestibility when calcium levels were increased in the diet. Okay, and I'll explain why that is a little bit later. Some of the more recent work, however, that we've done at the University of Maryland, and that was published by Kim et al. Earlier, well, actually last year, basically, at the Poultry Science meeting, I think that was, has actually shown that in some cases, with some phytases, calcium has no effect. So what we're measuring again is ileal digestible phosphorus in response to calcium, either 0.6, 0.8 or 1%, and diets with no added phytase in the red, or diets with added phytase shown by these gray bars. And what you can see is that increasing dietary calcium in this case suddenly had absolutely no effect on ileal phosphorus digestibility. And so compared to the previous work that I showed you with the E. coli phytase, or the earlier work that was published by the Tamman et al. Group, the question is, why are we seeing these differences? And one can really sort of summarize that by saying that in this study we were using a fairly high dose of a very efficacious Buddhioxella phytase and showing absolutely no effect of calcium level on the increment in ileal digestible P from phytase. Let me just move on. So really the sort of take-home message, if you want to avoid negative effects of calcium on phytate hydrolysis and also phytase efficacy, is first of all the rate of phytate hydrolysis by the phytase itself is going to influence how calcium affects the response. Okay, so phytases such as the Buddhioxella that work at an extremely rapid rate are going to be less influenced than phytases such as the E. coli phytase. The second point is you can also reduce the effects of calcium simply by increasing the dose of the phytase. And in this example here, again, we're looking at phytate hydrolysis in response to either 500, 1,000, or 1,500 FTU. And if you are rapidly hydrolyzing the phytate by using a higher dose of a phytase that is working at a low pH, must be said, the chances of calcium interfering with the phytase efficacy are going to be reduced. And then lastly, what I want to do is basically try and put phytate solubility and hydrolysis into a practical context. So obviously what we've just been talking about is the absolute rate of phytase hydrolysis as being important. Obviously there's going to be big species difference in terms of how phytases act, namely that in pigs you have a residence time at a low pH where calcium is not going to be influential, or at least less influential on phytate hydrolysis of about 3 hours, while in chickens we have a residence time in the gizzard proventriculus of about 40 to 60 minutes. So the importance of hydrolyzing phytate rapidly at low pH is much more relevant in chickens than it is in pigs, and also consequently there are going to be very different effects of different phytases in pigs and chickens purely because of the rate of, or at least the residence time in the acid stomach. And with that, basically there's just a sort of word of caution from a very practical note, is that some phytase suppliers have obviously not realized this or done their homework properly on matrix development for new phytases, because these companies are still recommending exactly the same values for broilers and pigs, which cannot be the case based on the different residence time in the acid stomach of those animals. So just moving on, I just want to really touch on a couple of other practical factors, I think, considered, namely the age of the bird that one's feeding is influential in terms of the matrix value, the substrate levels, as Dr. Corver mentioned, as well as thermostability. So the first question is, can one actually take a full contribution of phosphorus from phytase in a broiler pre-starter diet? I get this question a lot, especially from primary breeder companies who are worried, obviously, about bone integrity of the genetics that they are selling. And to answer that, basically, what's important to know, and the reason why the breeder companies are concerned about that, is simply that skeletal mineralization in this early phase, so early pre-starter phase, is extremely rapid. So in other words, in the first 10 days, you're getting about 28% bone mineralization, as opposed to sort of around about the 35% mark by 49 days of age or 50 days of age. So if you get it wrong in terms of your phytase application in this area, it is going to have severe downstream consequences for bone development and broiler performance. And that can really, again, be just visualized by looking at the rate of bone ash increase that one gets in these first 10 days. So the phytase efficacy in applying the right values in that age group is absolutely critical. And with that in mind, I was involved in some research at Scotthorst Feed Research back in 2012, where we specifically were looking at phosphorus retention in 7-day-old broilers to added phytase. And you can clearly see here that in the 7-day-old birds, the phytase response was really phenomenal in 7-day-old birds. In fact, it was even better in 7-day-old broilers than it was in 21-day-old broilers. And the P equivalence that we obtained at the time, this was Bodioxella phytase, was 0.156%. So the take-home message, in my opinion, is that provided the phytase is in the feed, there's no problem with adding phytase in terms of broiler pre-started diets. But having said that, what is absolutely important, as Dr. Corvus said as well, is knowledge of the phytate levels in your feed ingredients is absolutely super critical. You've got to know what the phytate levels are, and there's companies such as DuPont that do HPLC analysis of feed ingredients and provide tables of estimated phytate values in dietary as well as of grain that's ingredients, so I think those should be noted. So in summary, I think what really stands out is that one cannot generalize the phosphorus matrix value and say that all phytases were created equally. There's large differences between phytase sources, as well as obviously the dose of phytase and how they react in vivo. There's also large differences between phytase companies in terms of how they derive the methodology of the P matrix, which needs to be understood by nutritionists. And then obviously there's a lot of interactions in terms of dietary calcium level and phytate utilization, differences between the phytase sources being affected by dietary calcium, and then there's other factors such as bird age and passage rate that need to be considered as well. However, having said that, if the primary number one thing probably, we can go into as much detail on other factors that affect phytase use as we want, but if phytase is not recovered in the finished feed, in other words, it's not thermostable, then all these other factors are pretty irrelevant. And again, just on a practical note, in my experience, there are large differences between phytase enzymes that are on the market in terms of thermostability. And a take-home message really is that every single nutritionist should determine phytase recovery in every feed mold, because every feed mold is going to be different in terms of its characteristics. And with that, I'd like to thank you for your time and happy to answer any questions.
Thank you, Dr. Plumstead. Thank you, Dr. Plumstead. We'll now have our question and answer session. And our first question is for Dr. Corver. Dr. Corver, how important is the organic matrix formation at early stages?
I think that organic matrix formation is critical. That's the framework that really the mineralization is going to take place on. And so we need to have a proper formation. Essentially, the shape of the bone, which is going to then affect the structure and the function of the bone, is determined by the shape of that organic matrix. Now, as the bone develops, partially mineralized bone can be deformed by pressures. That was one of the problems that we often saw with heavy birds, particularly in the 80s and 90s, was the pressures just deformed that bone before it had a chance to mineralize completely. And then when it was mineralized more fully, it led to locomotion problems and other issues. Now the question is, what can we do to make sure that that organic matrix is formed properly? We haven't seen a lot of work on trace minerals and bone formation. I think we've pretty much been able to supplement diets with trace minerals that are involved in organic matrix formation without too many concerns of over-supplementation. So I think it really hasn't been an area of fruitful research, at least for the last probably 40 years even. Again, I think as we keep pushing these birds harder and harder, we may start to see issues of trace mineral nutrition becoming more important.
Okay, I have a question pertaining to the impact of bone strength at the processing plant. They ask, what's the value you need in tibia ash or breaking strength of the bone to avoid problems in the processing plant?
That's a good question. I'm not aware of any studies coming up with a threshold value. I think in general, the more mineralized the bone is at processing, the less likely you are to have a problem. I think that's one of the other consequences of reducing age at processing is we just do have a less mature bone. But unfortunately, I'm not aware of any studies that have related particular bone ash numbers to a relative risk of breakage at the processing plant.
Okay, thank you. This question's for Dr. Plumstead. What is your opinion about superdosing? I'm assuming they mean superdosing of phytase.
That's a real good question. Obviously, superdosing of phytase or high phytase dosing, as we call it, is a real hot topic. It's been a hot topic for several years. So I've been involved, I guess, with the commercial feed industry at least for the last 4 years or 5 years in promoting high phytase doses. All of the customers that I work with in South Africa have been doing that for at least the last 3 years. And I think there's no question, or there's a lot of evidence that it works. What is important, however, is obviously that the superdose that one uses will be dependent on the source of phytase. So there's no need really to go to a 2x or a 3x in terms of phytase dose to achieve the extra phosphoric effects for all phytases. So I think the important thing is, again, one cannot generalize that superdosing comprises 2,000 or 3,000 units. That will depend on the source of phytase and obviously also, again, on the sort of physiochemical characteristics of that phytase as to what effects one gets. But in general, I would strongly support the use of high phytase doses, not only because of the effects on amino acids and energy, but also from what I was mentioning in my presentation in terms of reducing or potentially reducing negative effects of calcium on phytase efficacy.
Another question for you, Dr. Plumstead. A couple of folks have asked, they said they understand that there are reference manuals that say, corn should have this much phytate in it and other feed ingredients. They want to know, are there any quick, easy ways for them to actually test the ingredients themselves or have them tested?
Unfortunately, not for phytate itself. There are some folks who are busy developing NRR-based prediction equations. I don't know how robust those are and whether those are really able to give an accurate, idea of what the phytate levels are in your feed ingredients. So NRR or NRT is definitely sort of rapidly progressing as being able to do that. But again, one would need to look at the actual sort of, how good those prediction equations are. The only other way is really to do a wet chemistry analysis using HPLC. But having said that, obviously phytase companies do that for their customers, and so I would just encourage the nutritionists to contact their phytase company and sort of kick that back at them in terms of providing the phytate levels of ingredients.
I have one question. How do you prevent the digestion of phytase in the bird? So
I'm sorry, could you repeat the question?
Yes. The question was, how is the phytase protected from being digested in the bird itself?
Okay, I understand. Thank you. Yeah, obviously, phytase is a protein, and it is susceptible to digestion by proteolytic enzymes. However, most of the commercial phytases that are out there have been developed by, first of all, screening the susceptibility of that phytase enzyme to pepsin and things like trypsin. There are differences between phytase enzymes in terms of how severely they are affected by proteolytic enzymes in the digestive tract. So again, I would encourage the nutritionists to ask for that data, or at least ask their phytase suppliers for that type of data, in order to ascertain themselves, whether that phytase company has actually done their homework and provided a phytase that is resistant to pepsin or trypsin degradation.
Okay, and have a question. Is it okay to use bone meal as the only source of phosphorus in a poultry diet?
Also, real good question. I don't know if Dr. Korver wants to comment on that. I think from my side, obviously the key concern about bone meal is the variation in calcium and P levels in that source. So obviously the percentage bones that go into bone meal tend to vary quite a bit, and the calcium and P digestibility can also vary depending on how that product is processed and how well those bones have been hydrolyzed in the cooking process. So personally, I would be, I guess, reluctant to use that as the only source of phosphorus in my diets. Again, totally depending on sort of what the quality is like. But in my experience, I'd be scared to do that.
I agree with Dr. Plumstead. I think the consistency issue is very important. If you're formulating diets assuming not only a level of calcium and phosphorus in the diet in total, but also the relative availability, if you've got a consistent source and you're comfortable with it, that's probably important. If you're not sure of the consistency, you could run into some very serious problems.
Can high phytase inclusion interfere with calcium absorption or deposition?
That's also a real good question. No, I think basically what high phytase inclusion does is it will hydrolyze all of the phytic acid in the acidic portion of the digestive tract and really make calcium more digestible. So what needs to be done is the calcium levels of the diet need to be adjusted downwards when you are using high phytase levels, and that will also obviously depend on the source of phytase. So provided that you are adjusting the calcium level of your diet when using high phytase doses, I don't think that there would be a problem in terms of calcium utilization. If anything, it should increase. What one does need to be very careful about, however, is there's still folks out there who are not adjusting the calcium levels of the diets when they are adding high phytase doses, and then you certainly can run into problems in terms of a calcium P imbalance.
Now we've got a question regarding layers, a little off the main subject, but it says there are several phytase suppliers that still recommend lower doses of the enzyme for layer diets, even if the soluble phytate content is high in those diets. And they ask what you think about that.
Again, very, very good question. I've been involved in some Langhen research, and from what I'm aware, DuPont is also doing some work directly with Dr. Corver in his lab using the computed tomography to actually look at laying hens over their lifetime. And I disagree with using or continuing to use a very low dose, in other words, the standard 300 FTU unit in laying hens. There's no question that the efficacy of a phytase in laying hens is higher than in broilers. In other words, you don't need quite as much. However, in my own research that I was doing while still with DuPont, we were seeing very good responses to phytase in laying hens, right up to around about 1,000 units. So I don't think that there's any reason to continue to only sort of restrict oneself, I think, to a 300 unit dose of phytase in a laying hen.
My impression of that is one of the reasons I think we've used lower doses of phytase in laying hen diets is because with the mature digestive tract you do tend to get a higher inherent digestibility of phytate or higher available phosphorus, and I think in the past it may have been a cost-saving effort by reducing the phytase supplementation.
What is the maximum phosphorus from phytate that can be released by the use of phytases?
I see we're getting some real good questions coming through today. Also, yeah, very, very good point. Typically, again, there's research data and then there's a practical element to that answer. So in our research data, we've got really good studies that show that we can liberate from 1,000 units of phytase sort of right up to the 0.2, 0.22 available P contribution, which was, those diets contained approximately 0.26% phytate. So we're getting up to sort of the 80–85% mark in terms of phytate P utilization. However, having said that, obviously from a practical context, one always needs to be conservative as a nutritionist. And the advice that I give folks out there, at least commercial nutritionists, is to stick to round about the sort of 67 to 70% of the phytate P in the diet as an absolute maximum cap on a phosphorus matrix value. And that's another reason why one obviously needs to be aware of the phytate P levels. And again, that will obviously again depend on the source of phytase that one is using. But for the new generation phytases out there, I would still recommend keeping it to the sort of 67 to 70% of dietary phytate as a maximum limit.
Okay, one last question. And yes, what are the most important factors that have an impact on phytase efficacy?
I think it's difficult to rank the factors. I think but calcium certainly is probably the number one sort of influence in terms of phytase efficacy. And you can pretty much, again, hard to generalize, but effective as a mutation will totally depend on what phytase source and what dose of phytase one is using. So again, with your new generation phytases such as Bordeoxella that work very rapidly, calcium is less influential. Aside from calcium's phytase efficacy, obviously the phytate level in the diet is going to be critical, as is the fact that phytase needs to be in the feed. So probably point number one is phytase needs to be in the diet, and one needs to be able to measure that, and then look at the calcium levels and look at the phytate levels as probably the top three factors.
Thank you. I'd like to thank all of our attendees today for taking the time out of their schedules to participate in our webinar. A recorded archive will be made available at www.wadagnet.com/webinars.astx. Today's webinar will be available to be viewed within the next few days. Please don't forget to take a couple minutes to answer our short survey at the conclusion of our webinar. Remember, we have several upcoming webinars and webcasts, including 2 this week, webcast on Thursday, and then webinar on Friday. And you can go to our website and register at any time. And I'd like to thank DuPont for sponsoring today's webinar. For more information on products and services offered by DuPont, please visit them at www.dupont.com. And of course, I'd definitely like to once again thank Dr. Korver and Dr. Plumstead for their very interesting presentations again. Thanks to everyone in the audience, and I hope you have a great day.